All right, so this is the last video I'm going to do for today. I want to thank everybody who's actually commented on to me about them. I'm going to continue to do them for myself because I think it's helping me communicate what I want to, but I'm happy to have people listen along too. So this is just a few slides, I think like 4 maybe, on transcription and translation and why this is really important in the cell, and then also for viruses. And so I'm going to just maybe talk through these. I just have some pictures and we'll approach it that way. So what you're seeing here is a cell. And it's a cutaway of a cell, of a eukaryotic cell. And so over here is the nucleus. And in the nucleus, we see all this packed material. And this is chromatinized DNA. And in this case, it's sort of unwinding a little bit. And this is what actually allows us to go from having DNA to a fully functional protein, which we talked about last time, like hemoglobin. And so how does that occur? Well, there are these enzymes here, which are made up of protein and RNA, and these are known as polymerases. And in this case, it's an RNA polymerase. And so the RNA polymerase, along with a host of other proteins, will access the DNA and to get to those base pairs, which we talked about. And it's a really neat system. What it does is it goes and it reads along, and it reads each base pair. And it says, okay, here's an A, here's a T, here's a G, here's an A, G, C, whatever they are. And it will make a complementary strand of that from that DNA, and it'll make it into RNA. And so it's reading the DNA, and whatever that base pair is, it's putting the complementary nucleotide to it. The exception here, I won't really go into the details, but one of the bases used in DNA isn't used in RNA, and instead a different one is used. And I don't want to confuse anybody on it, so I'll just leave it at that. It's not 100% the same, but the message gets read the same nonetheless. So you start with DNA, you make a message, and now you have what we call a messenger RNA, and that is this thing right here. This is the message, and this is going to tell the cell what amino acid it should make based on the configuration of these nucleotides in this messenger RNA. And so these are ribonucleotides, right? Because they are not DNA anymore, they're RNA. And so now you have what we call a ribosome. And the ribosome will dock onto this messenger RNA. And again, there's so many details here. I'd love to go into them, but I'm not going to. But there's a lot of proteins and factors involved to get this to work. But what it does is it starts adding every 3 of these bases encodes for 1 amino acid. And so it starts adding these amino acids, and it makes a string of them like I covered in the other video, the previous video. And we can see them right here. This is 1 amino acid, and then here's another amino acid. So here's the nitrogen, and then that's the carboxy end, and then we're back to a nitrogen. And so you get this string of amino acids, and it starts to form its secondary structure, which we can see right here through this alpha helices. And these all come together. We've got these ribbons here. These come together then to form that tertiary structure. And then this is our fully functional protein. And so now when you think about viruses or even cellular proteins, when somebody says, this virus is making a protein, you have some sort of sense now as to where that protein came from. It didn't just appear from thin air. It started as some genetic material, whether it was the cells or it was the viruses it brought in. Let's say that this thing right here is the virus, the genetic material. And it has its own polymerase, and it makes its own mRNA. And then it makes the cell make its mRNA. It steals these components from the cell, and it makes it do its work for it. So yeah, I think that's really cool. Here's sort of a close-up image of what that might look like, a model. This is the DNA, and in the orange, we've got the this polymerase, and then this green here looks like it's supposed to be the messenger RNA, but you can see it's opened up the DNA right here. This double helix is opened up, and it's reading through rather, and making this messenger mRNA. And so we call this then the essential dogma of biology, and that is that that biology progresses from DNA to RNA to protein. as with many things in biology, there are exceptions, but if you just take this for sort of your baseline knowledge that DNA goes to RNA, goes to protein, and not the other way, unless we're talking about the exceptions. Couple other factoids: DNA is read by those polymerases. So DNA-dependent DNA polymerases for humans, or for animals, or any cellular life for that matter. And that makes the messenger RNA. That mRNA is the message which encodes the protein. And so the ribosomes then translate that message, and that makes the fully functional protein. This is, again, just another cartoon showing that progression of DNA, and then the polymerases would read through this and make this messenger RNA, and then ribosomes would dock onto this, and they would start making this protein. And so that's how proteins are made. Now, when a cell needs to make its own DNA over, it does that through, not totally, it's a similar fashion in that the DNA polymerases for a cell would open up this double helix, and it would start synthesizing a strand for each of these. And so you wind up with two new DNA strands. Well, you wind up with two DNA strands, and each of them has one of the prior DNA strands as part of it. And the other one is the newly synthesized one. And so we call that semi-conservative. This double helix would get opened up, and now you've got sort of like this scissor sort of configuration. And on each of those open strands, you would start a polymerization of new nucleotides. And so at the end, you have two DNA double helixes now, and one of those strands of each double helix came from that parental strand. And this is just the cell's way that it's evolved to easily and unmistakably make and replicate its DNA. It has proofreading mechanisms that can go back through and read through that message to make sure, read through that sequence to make sure that all the A's match to the T's and all the G's match to the C's. And it's interesting because not all viruses have that, especially like this new SARS-CoV-2. It does have a proofreading mechanism, which is kind of unusual for RNA viruses, but it's not as good as the DNA one. Something like influenza virus doesn't have it. And so that's why you wind up with these RNA viruses, which people talk about, which are really sloppy and they mutate fast. That's because they don't check their DNA, or their RNA rather, when it's done being made. And so again, I'm a big proponent of showing you and trying to get you to understand that, like, you don't need to take my word for it that these things exist. We have electron microscopy that shows us these things exist. And so that's how we come up with our understandings and hypotheses based on that. So an electron microscope, you coat your sample with some sort of electron dense material. So in this case, maybe gold. You can coat it with a coating of gold, and you bombard it with electrons, and you wind up with this image. And what you're seeing here then is a strand of DNA, and where transcription began, right? And you've got these transcripts that are coming off of the DNA now. This is where the polymerase was reading, and it's making those mRNAs. And then over here, they've marked these ribosomes. So not only did they capture the mRNAs, but they've captured ribosomes actually making the functional protein. And then for this example, this is what is sometimes called theta replication, but this is probably a bacterial plasmid or maybe a chromosome. And you're seeing unlike humans who have two sets of 23 chromosomes, a lot of bacteria only have one chromosome, and it's a circular chromosome. And so what you're seeing here then is the very beginning of its process of replicating. And so it's opened up this origin, it's opened up its double helix of DNA, and it's going to start synthesizing a complementary strand in both directions. And given enough time, this is what it looks like. You have two of these circles now. And with the help of some other enzymes, it'll eventually cleave those off so that you wind up with two independent, separate circles of DNA like this. And so I think that's really cool. We have models like this that we can show people, and people, you might take it for granted, or you might take somebody's word for it that is what happens. But you don't need to, because this stuff is out there. This is available for you to go and look at and say, oh, wow, there's actually, this is something we visualized. We didn't just need to maybe assume it, or it's some hocus pocus magic science. Like, we actually see this happening. And so I think that's really important to getting over that hurdle of maybe people don't trust scientists, or they don't understand it, therefore they don't trust it. It doesn't need to be that way. So yeah, with that, those 3 major videos, I think it's a lot of information. It's clearly, yeah, it's a lot of information. So if you have to listen to it a few times, or if you have to ask questions, I'd be happy to answer when I can. Moving forward, though, I'll start focusing on the immunology and the virology side of things. So I look forward to doing that. All right, thanks for listening.